So thank you and joining us for the BrainX Community Live for the August 2020 session. And just like a brief history about BrainX community, we have been having 2 successful years of various educational sessions, our focus being on machine learning in healthcare applications for good. 2,000 plus international membership now, and then various forums for us to get together and learn and exchange information and collaborate at. Again, a reminder, this is the web page. We started with 3 sections, but now we have grown to like multiple more. The Connect one, where now we are online. We went to Zoom earlier this year. The YouTube channel that we started, where this presentation will be available later. The very active LinkedIn group. And then the Learn section, where there is a list of curated and annotated articles for machine learning in healthcare. The data section, which has been increasingly expanding, and I'm glad that a lot of groups are sharing their data and making it available open source. The COVID-19 dataset especially, we released our dataset last month, which was the collaboration between BrainX and TAF Pritesh Sethi in India, interestingly curated by 2 high schoolers. And then the BrainX community that was launched last year, the journal section, meeting section, which has been ever expanding and going to virtual. And here is an example of Transcontinental Collaboration, something that we published a few months ago. And then again, back to the BrainX community with the various forums. So today, we are blessed by having Dr. Lara Jehi. She is the Chief Research Innovation Officer for, or Chief Research Information Officer, my correction, for Cleveland Clinic. She's a neurologist here, and she is also very well published. And this session will be moderated by Dr. Kamal Maheswari, who just joined us. I'm going to give him a moment to join.
Hi, Piyush.
Hi, Kamal. So Kamal is the medical director for Center for Perioperative Intelligence, and he will be helping us moderate this session with Dr. Jehi and about a research paper and various aspects of data analysis and machine learning. I'll give it to you, Kamal.
Hello, everyone. Thanks for joining, and especially thanks for Dr. Lara Jehi for giving your time. I know it's busy, and it's always, the activities in this area is more towards the innovative side, and some people can call it hobby side, but this is all what we are passionate about, what we're trying to do. Once again, thanks a lot for joining. For everyone, Dr. Jehi is a neurologist, and she's specialized in epilepsy, and she's Chief Research Information Officer for Cleveland Clinic Health System. She's also a Vice Chair of an Institutional Review Board and a Principal Investigator of Cleveland Clinic Biorepository. She have hundreds of publications, what we're going to specifically focus on today is a recent publication regarding COVID. The COVID, there's a huge amount of information which is coming out, and to make sense of the information from COVID, within Cleveland Clinic, there's a COVID repository or data registry. And using that, the question which was asked was that which patient should be tested, and what is the value of a test in a COVID, given the scarcity of the testing. I would let Dr. Jehi explain the paper in full, but before she describes the paper, I would also urge her to briefly introduce her role as the Chief Research Information Officer. Dr. J.
Yeah, thank you. Thank you, Kamal, and thank you, Piyush. This is I've been looking forward to this meeting to get to know the group and learn from everyone. I've been in a Cleveland clinic since 2000. I initially came to the clinic as a trainee in neurology residency and then stayed on for fellowship. I wore various hats, but then the last one was this role as the Chief Research Information Officer for the organization, which is a new role for Cleveland Clinic. Nobody held it before me, so I had no template to follow, right? That's could be a good thing, could be a bad thing. The good thing with it is that the slate is clean and we can create something new that's going to help the organization. My vision for it is that my job is to make sure that the infrastructure is there to enable research and through that advance innovation and science, right? So both business development and basic science, translational science advances. So I had just been in the job for a month and then COVID hit. So all of, all of the time that was initially planned to go into learning, current state, learning the needs, taking my time went away. And really we had to respond as an organization to COVID very quickly and come up with an information resource infrastructure that can help us answer the scientific questions that people were interested in. And I have to say that the testing, the drive-through testing started at the clinic on the weekend that was around March 17. Mm-hmm. March 18, we had the IRB approved for the COVID Enterprise Registry. Initially, with just 4 people on the team. Within a week, we grew that to 24 people, physicians, scientists on the team, and we had 4,000 patients enrolled. I will show you, some of those numbers as this progresses, but in effect, it was an exercise of what can be done when a team is built and people collaborate towards a, common goal. One of the first questions that we wanted to answer is what we did in this paper, which is what's the likelihood of people testing positive for COVID based on their symptoms and comorbidities and other factors. Basically, what we did is we mapped the journey of a patient with COVID. Testing is the first step. Second step is if they need to be hospitalized. Third step is progression and disease severity, etc. And we collected data and built our models to predict proactively each one of those steps. Let me share my screen now. So I start showing you the data related to that paper, and then we could spend some time discussing afterwards. So let's see, it's not advancing, it should advance, no? Okay, yeah. So the first question is, why did we build this diagnostic discrimination calculator? Why was there a need to have a model out there that can predict whether a patient is going to be positive to have COVID or not, right? So the clinical challenge is that testing resources for COVID-19 do not match the need. They haven't matched the need in March, and they still do not match the need now in almost September. And the expert opinion recommendations are imperfect, on who needs to be tested. And there is no better evidence for that beyond the never-ending changes that we keep coming up with for who needs to be tested, right? Symptomatic, not symptomatic, older than that age, younger than that age, and those criteria keep changing. because really there is no real science behind them. So the aim of the study was primarily to develop a tool that integrates the patient's clinical symptoms, exposure, medical comorbidities, vaccinations, and current medications to generate this individualized risk of the likelihood to have COVID. We included things like medication history because we worked with teams within our computational science group in the Lerner Research Institute, and they had done a lot of work on drug repurposing studies, and they had identified a list of 16 medications that potentially could have some effect against COVID. So we made sure to include those in the registry up front to see whether they influence risk in any way. The secondary aim was to use what we learned to try to identify some potential mechanisms of disease susceptibility. How did we answer the question, right? How did we develop this tool for individualized risk prediction? The patient population was basically everyone who was in the registry at the time. It was patients of all ages tested for COVID in the Cleveland Clinic Health System. So that is all of our facilities in Ohio and our Western facility in Florida during a specific time frame. We had close to 12,000 patients who fulfilled those criteria, of whom there was 800 or so who were COVID positive. We had close to 300 data points on each individual, but then ultimately we identified those that were most relevant for the predictive model, which we then validated in a cohort that was tested in Florida. So we wanted that geographical distance. We built it in Ohio, we validated in Florida, and in a different time frame. So 2 weeks after the testing set was done. And what did we generate with it? You have the reference for the paper first. From a practical standpoint, though, what it generated is this online risk calculator, which is publicly available, where a physician can enter a patient's age, race, ethnicity, gender, smoking history, BMI. The zip code is there because we also included social determinants of health as being a group of variables that we wanted to evaluate, and they actually ended up being quite relevant in the prediction of test positivity. Symptoms, comorbidities. So the zip code links us to about 4 measures that reflect the disparities. And then the vaccination history, the two ended up being relevant, actually, the pneumococcal vaccination history and the influenza vaccine. Medications, and then based on that, you could run the calculator and then get a patient's individualized risk of having a positive test. How good is the model? You see the numbers there. The C statistic in the development cohort was 0.86, it was 0.84 in the validation cohort, and the calibration curve on the right shows that it performs okay. And then the last question that reviewers wanted to see was some clinical interpretation of that individualized risk that we come up with. Like you saw in the example that I showed, that particular patient had a 14% risk, right? So what does that mean? Is that too high, too low? What do we do with it? And for that, what we did was build this curve that plots the proportion of negative patients that we would avoid testing versus the proportion of positive patients that we would detect throughout the predicted probability cutoffs, meaning, and then for each cutoff, we calculated sensitivity, specificity, negative predictive value, and positive predictive value. For a clinician, this is particularly helpful because they don't want to go thinking through all of what risk means and, you know, how do I take that number and respond to it based on epidemiological data. That's too complicated. They just want now something that says, this is high, this is low, this is medium. And, you know, then they decide what to do with it. They preferably want a recommendation for what to do with it beyond just this is high, this is medium, and this is low risk. So this is what a plot like this can help with. And we ended up going by the point where both curves intersect, meaning you would be detecting as many positive patients as you would be avoiding negative patients. And that cutoff was 12.3%. The clinical applications for this have been quite interesting, actually. So after we wrote the paper and spoke about the calculator, then people started coming to us with uses that they saw for it. So we adapted it for them. One use is in the home monitoring program that we have going on now in the enterprise, where every patient who tests positive is enrolled in this program where a case manager or a nurse contacts the patient every day for 14 days after the result of their test and goes with them through a survey of symptoms to help detect whether they're getting worse, staying the same, you know, or progressing. So since there are patients who have symptoms of COVID but don't quite fit the criteria that warrant testing, right? It's because we want to preserve testing resources. So like younger individuals, for example, they would just be given the clinical diagnosis without ever being actually tested. And those people fall under the category of suspected COVID. And they were given the same, you know, let's call you every day for 14 days deal. When in reality, we know now, and we've known for a while, that most people who have COVID do very well with it. And it's 20% of all those infected, we think, who progress to severe disease that requires hospitalization. So for this group, what we did is we integrated the calculator within our electronic health record system, which is Epic. We also integrated another calculator that we developed right after this one that predicts risk of progression to severe disease in those who are infected. And what happens now is the program on the back end from the electronic health record can calculate an individualized risk and score for each patient for their likelihood of having COVID and their likelihood of progressing to hospitalization. So the home monitoring program now can sort all their patient lists. They have thousands of people that they follow by those scores and then prioritize those at highest risk and make sure to monitor them closely so they don't get lost in the mess of, you know, all 100 who may do just fine. So that had a good use in that it was prioritizing clinical care to those who need it the most, and it was helping with manpower allocation. A similar application to this is happening in the emergency room. So those two, so the paper I talked about the most is the test positive calculator. There's another one, like I said, about the hospitalization risk. And Those two are just examples of the data research projects that we were able to accomplish through this COVID-19 registry that is now interfacing with the biorepository for the clinics. That's allowing us to phenotype COVID patients that we're collecting samples on, and with our clinical trials that are ongoing. This is the enrollment of patients in the registry, and you see that now we are close to 30,000 or so people who are in it, and there is that wealth of data on those patients. So what started off initially as a project that is designed to help us answer these critical questions of who tests positive, who is going to be hospitalized, ended up being this resource that is now being used by hundreds of research teams, as you see there. We opened up a call for research ideas. We did that very early, within the first week actually of starting the registry, because we wanted to make sure that we adjust what we collect to the needs of the community. What are our scientists interested in? So we ended up with a database that has a bit over 300 data points on each patient. And still, that's not everything that people were interested in. We had to draw the line somewhere. But it's quite a heavy data set. We ended up consolidating a lot of those teams because you can imagine many people were interested in the same things. We didn't want them to duplicate, so we put them together. That went from 208 down to 143 groups, and then prioritized those who had clear, really clear questions, and we've fulfilled their data requests first. And so far, I have more than 30 because I really don't know the exact numbers. I know that the clinic investigators have published 306 papers so far since March on COVID. They're not all obviously from the registry, but I know of at least 30 of them that are their papers in journals like JAMA, Lancet, Chest, what we have here. And there's a lot of grant funding that went in using this resource. So altogether, what I learned from it is that a lot can be done when it's done in a thoughtful way, meaningful way, and with a purpose, rather than, so you start with the purpose and you build the data towards it, rather than, we have a lot of data, let's see what we can come up with, kind of approach. And with COVID, the purpose was pretty clear. I will stop here and we can open the floor for questions.
Great. Thanks a lot. This was very insightful. It is impressive that the foresight which went into developing and this registry at such earnestly and so quickly, and this kind of collaboration can happen in an institution like this, and that's amazing. So first of all, congratulations on leading this effort, and it's quite impressive. Secondly, I like the idea that opening the problem solving or the problem to the wider community. So not only that we, every person have one or few ideas which they want to deal with, and opening it to the broader community will maximize the use of this data for broader good. So I really like that. Specific, now I have one question for specific to this publication or this registry use, and then there's some few questions which are there on the chat group, and I'll bring it up to you, okay? Mm-hmm. The goal here for this risk calculator, was to maximize the negative predictive value, right? Or to an extent, so correct me if I'm wrong, and then that's for the discussion which if the patient have risk factors, you apply this calculator, and if they don't they are not likely to be positive, we can save some tests?
It depends on really. I've been amazed by how people want to use this. So
Yes.
there is so Epic, right? National
Yes.
Epic Wisconsin. We've been they heard of this, they saw it. So so we built a version of it for them. And what they want to use it is actually the other way around. They want to use it where instead of testing everyone every day, they are just going to be using the symptom checker and some rough predictors otherwise, race, gender, smoking, to identify then who is high risk. And then they're talking about, the vision is use it in big places of employment where instead of the, like the thermoscan thing, people can take this, and if they are high risk, then those would be the ones they would push aside and then, maybe send for testing. So it is, so for them, it is really a screening tool. It is not let's save resources and not spend money on testing. Others are thinking about it that way. I have pharma companies who want to use it as a way to identify people at highest risk for having or catching COVID, so they enroll them in vaccine trials, right? So depending on what problem you are wanting to solve, you will choose a different cutoff for where that prediction needs to lie, and you could adapt it.
Yeah. So, and the reason I asked the question, because the positive predictive value was at multiple level was on the lower side, right? So if you want to hedge the risk, you go on the flip side, which was the safest bet. That's what I was trying to get at.
Correct.
Okay. I do have a question regarding first question is from Avirup Guha, and this is more about model feature selection. So there's, in machine learning to our routine statistics, which I can call it routine statistics now. The feature selection is very important component for any prediction system, right? And people use different technologies or different methods. So we use Lasso ourselves a lot, and there's random forest. So is there any particular reason why you choose?
It just provided the best discrimination. So Mike Catan's team in QHS tried different models to see which ones would have the best discrimination and the best calibration, and Lasso did the best. So that's why we went with it.
Okay, so thank you. The next question is from John Lee. Did your model result in reduced testing volume? In particular, did you reduce any pre-procedural testing?
No. No, it didn't. The pre-procedural testing decision was made because of operational necessities, in the sense that even though the risk of we followed that data through the registry, and it's been steady, I would say, for several months now, that the proportion of people who test positive out of those asymptomatic being tested before procedures hasn't really gone above 0.6%, been somewhere between 0.4 to 0.6% of people who have no symptoms get tested. Only those are positive. So it's a very small proportion. But the model was developed on those who had symptoms and got tested, right? So to start, so first, I cannot say that, with a straight face, that this model is good enough to be applied on asymptomatic individuals, and it would predict their risk of having COVID. That's a leap. We would have to validate it in that population, which we're looking at now, but I doubt it will do well, because with such a low proportion of people being positive, I just don't think that any statistics will be able to find them. So then it boils down to what would be the consequences of missing 0.5% when you have a surgical volume like we do, who have COVID, and
exactly.
you have it unless you test them. So that is a purely, operational healthcare management question, and it's not one of statistics and
Okay. Yeah. No, it's a hard question. Hopefully, John got the answer right there. And there's a follow-up. Okay, there's a question or a comment from Piyush. Where do you see research, machine learning, and IT intersect, in your opinion, in the new role as a Chief Research Information Officer?
I see a big intersection. I would say we should talk. So, so in that vision that I was talking about, with infrastructure being there as a requirement, but really the goal is enabling research so that we can drive that innovation and drive that progress, that scientific progress that we are talking about. That vision is agnostic of a tool, right? So it could be traditional statistics, if there's anything that could be called that. It definitely should include artificial intelligence and machine learning and multimodal predictions, and we shouldn't spare a way, right, to achieve those goals, keeping in mind that at every step of the way, we should always ask ourselves, well, what's the best way to answer the particular question, right? So some questions may not fit well with machine learning, others may do, right? So I think it's too absolute of a question to say, should we use it? Of course we should use it, but the answer depends on how, where, and when.
Yeah, no, it's absolute. I think whichever method fulfills the purpose and makes the best results, should win, like anywhere else. One question from Srinivas, congratulating you for a Herculean effort, and any comment on using Bayesian approach, for example, utilizing the physician clinical gestalt as a prior informative. I'm trying to read the whole thing so that I'm not misrepresenting any facts here.
Okay, yeah, so let me
and of course, obtaining the clinical gestalt would be difficult given the challenges of practical NLP tools available to.
Yeah, so this is very nice. So this is Srinivas. Okay, yes. What does Srinivas do? May I ask?
Hi, Laura. I'm Shri Mamadi, one of the pulmonologists and one of the medical directors for clinical informatics. I think I was there in one of your phone calls a couple of months ago or 3 months ago. Again, great work. So nice to see all this getting executed. So
thank you. It's a great question. And I don't know if the call you were on was the one we had with the emergency room team, but was it? There is a project that we have going on where we are trying to answer exactly this, where we are comparing the prediction of the model with the clinician gestalt, and we're doing that in the emergency room because it turns out part of their routine clinical workflow is that the physicians there have to provide a clinical gestalt judgment for high risk versus low risk of a patient to have COVID, depending on their clinical picture. So it's that validation piece is critical, and it's important to do it. So thank you for asking that question.
Okay, so great. I don't have any more questions, but Piyush, you have any comments?
No, I think this was a great discussion, and Dr. Jahi, thank you so much for presenting this. I think this was very, very helpful to a lot of members, and I think it was very insightful. I think it was very insightful, and your openness and your ability to share and promote this culture of research and being able to engage other people is something that we promote in BrainX community. Mm-hmm. So I think they go hand in hand with this. and some very good discussion that took place today. So thank you very much for that. And thank you, Kamal, for moderating this session. We are a little bit over time, and I appreciate everybody sticking around. But thank you, everybody, for joining us.
Thank you.